So I will now hand it off to our very first speaker. And that speaker is, I know we have all the speakers lined up here. Who is our first speaker? Rong Fan.
Rong Fan. Hi, Jim. Thanks. Okay, I'm gonna go ahead and share my screen. Got it. All right. So thanks, Jim, for the opportunity, and also the Cancer System Biology Consortium for kind of supporting this workshop, this symposium. I think that this is a great opportunity for us within this community to kind of share notes, to compare notes, and also kind of work together to move the field forward and to really have this new tools, either sequencing or imaging-based spatial multiomics tools to drive the discovery in cancer system biology. So I'm gonna focus on the sequencing, or more precisely speaking, NGS-based spatial multiomics mapping. So this is my disclaimer. And so I think Jim gave a very nice overview of the technology landscape, including the history, the early landmark papers. I often use this kind of diagram, or this tree diagram, to describe the technologies in this space. And I tend to use the word nowadays called it the family of the technology rather than the landscape, because often we need a multiple tools, technology to work together to understand better the cancer biology and the systems level. And broadly speaking, there are two main directions of the technology innovation in the space. One is based on imaging. I think you will hear from other speakers. And the other direction is what I'm going to focus on here in my talk, is using high throughput NGS sequencing to do spatial kind of encoder analysis of either gene expression or other type of omics. And the history of NGS-based spatial omics really start with this landmark paper published by Professor Joachim Lomberg from StarLife Lab in Stockholm. And he used the DNA microarray to capture messenger RNAs released from permeabilized fresh frozen tissue section you place on top of the DNA microarray. And messenger RNA is going to diffuse out of the tissue and captured by the DNA microarray. Each spot of the DNA microarray has unique sequence to encode the spatial address, and such that when you synthesize the cDNA, eventually you're going to incorporate the spatial address code into the cDNA. And when you do parent sequencing, you're going to read out the address code. You're going to read out what that gene is. That's the fundamental principle of this spatial encoder, spatial encoder whole transcriptome sequencing. So since then, there are many different technologies developed along the same line. So barcode solid phase substrate for messenger RNA capture and the spatial transcriptome sequencing. As shown here, you can see the spots are getting smaller and smaller, and also the tissue, the types of tissue that can be profiled using this approach is no longer limited to fresh frozen. That can be used for FIPE. And in particular, during a couple of years, the spot size even approach the subcellular. You are able to do sort of near optical resolution for spatial whole transcriptome sequencing. Since I'm speaking here, I'm going to highlight some of our own work in this space and what is unique and what is well differentiated in this whole species. We don't really take messenger RNA out. Instead, we send the spatial address codes into the tissue. The tissue itself is in the reaction chamber. You can perform a variety of different chemistries to look at the gene expression in the transcriptome. You can look at the proteins. You can also look at many other mechanistic aspects of the gene regulation, for example, epigenetic, chromatin accessibility, the histone modification, and so on and so forth. Or you can combine them to realize spatial multiomics. That is kind of the focus of the workshop today. And so back to this landmark paper published by Joachim Lomberg, and later they published this Nature Protocol paper. If you really want to dive deeper into how the technology works step by step, it's a very nice read. And also in this paper, they really coined this whole approach called the Barker solid phase RNA capture for spatial transcriptome profiling, and many other technologies that developed over the past couple of years really follow this fundamental principle developed by Joachim Lomberg. And one example is the SLISIC. Instead of using the pink spotted DNA microarray, they use the beads. You pack the beads in as a monolayer, and every bead has unique DNA barcode on the surface, and the bead monolayer becomes the DNA microarray. You can use the beads to capture messenger RNAs and perform the same chemistry, just like what Professor Joachim Lomberg developed. And then you can approach almost like a cellular resolution spatial transcriptome sequencing. So this is now commercially available by the company called Curia Biosciences. And so, over the past 2 years, and people further pushing the boundary in this technology innovation, and 3 papers came out within like a 12-month of time frame to demonstrate you can get this resolution approaching almost like an optical microscopy resolution because the spot size is as small as like a 1 micron, even smaller. And just to show you one example called a 6-scope, that's the first of the 3 papers, all of them published in Cell during 2011, 2021, or 2022. And what the 6-scope does is to utilize kind of the standard flow cell of DNA sequencing device, like a Illumina flow cell. And once you use those kind of random primers to generate the colonies in a Illumina flow cell, and the colonies you generate in the flow cell serve as the DNA microarray. Once you sequence those sequences, you know exact barcodes you have for every single colony spot. And now that becomes your DNA microarray. So you can apply exactly the same chemistry developed by Joachim, demonstrated by SLISIC, and other methods to do the spatial transcriptome, the spatial messenger RNA capture, and the transcriptome sequencing. But what is much more advantageous in this case is the array spot size can approach almost like a 1 micron, even smaller, such that you can resolve not just a cellular or near cellular level, you can actually resolve subcellular, as demonstrated here, the nuclear versus cytoplasmic RNA molecules. You can approach sort of subcellular spatial transcriptome sequencing in an untargeted, unbiased base-by-base sequencing manner. So just a couple of weeks ago, I think 10x Genomics launched this high definition or high density spatial transcriptome sequencing. I don't have to give an introduction about how the vision works. That has been widely adopted nowadays, and then based on Joachim Lomberg's spatial transcriptome DNA microarray approach. But recently now that they are able to make the spot size about 2 micron spot size, but typically you'll be 8 by 8 by 4 by 4, you get a 8 micron beam size as the standard practice for high resolution spatial transcriptome. And what is kind of worth pointing out in this case is it works quite well for even FFP tissue samples because 10x Genomics developed a huge panel of the probes to target almost the entire transcriptome for human and also for mouse. And so now in this case, you're now directed sequencing base by base, the whole transcriptome. Instead, use those probes and there's 2 probes to hybridize to the same target, and then you ligate the 2 probes together. If they're in adjacent, right next to each other, you are able to ligate them, and then you're going to join the 5' array sequence and a poly A tail. And then once you release this probe, you can use the same chemistry on the same device, use for the kind of the poly A messenger capture device to capture those probes, and then you'll synthesize the complementary DNA out of those probes and the sequence to do kind of genome scale, in this case, over 18,000 genes for both human and a mouse tissue specimens. So this is a kind of nice example. You can see significant resolution increase and as compared to conventional Visium or other methods. And if you're interested how that worked out, I think this kind of, if you go to the 10x Genomics website, you can look up some kind of technical technical specs, I think, for this 10x Visium and the Visium FFP and the most recent Visium HD. All right. So now I would like to really give you some idea about the technology we developed. And so we published this work back to 2020, showing for the first time you can combine the whole transcriptome and a panel of proteins for spatial multiomics sequencing. So honestly, the first demonstration of spatial multiomics sequencing. And so that time we mapped out about 20 to 30 proteins plus the whole transcriptome spot by spot at a spot size close to 10 micron. And so later we were able to use this in-tissue barcoding approach to map kind of epigenome and a multiomics. And so this turns out, indeed, this in-tissue barcoding concept is very versatile. And that's the unique advantage here. And since we published the work, I think many other approaches that also kind of try to adopt this in-tissue barcoding rather than capturing approach to do spatial transcriptome or spatial genome sequencing. The first one was from the called Tripner at University of Washington. They used the hydrogel DNA spot hydrogel to kind of transfer the DNA barcodes into the tissue. And then later in the slide DNA sequencing, and just a couple weeks ago, a couple months ago, the slide tag kind of modified from the SLISIC. They don't actually use the bead array to capture messenger RNAs anymore. Instead, they use the photocleavable DNA barcodes on a beads, and then the photocleave the barcodes, the DNA barcodes, release them into the tissue to perform in-tissue barcoding for either genomic DNA or for the transcripts. And so it turns out that's a very versatile approach. And when the spatial transcriptomics was highlighted by Nature Methods as the method of year, and they also wrote a small kind of research highlight for our paper saying multiomics sequencing goes spatial. And so that's the first demonstration of spatial multiomics sequencing. But I have to say that time we mapped out about 20 to 30 proteins, which sounds pretty cool. But probably that doesn't really meet my standard to call that multiomics or omics. And then I kind of try to challenge my postdoc, my students, can we really map down hundreds or even thousands of proteins to really approach kind of proteome-wide mapping and in conjunction of whole transcriptome. Turns out that's totally durable. But of course, how to build such a large panel of antibody DNA conjugates is a lot of work, but technically it's totally durable. So we demonstrate that works for about like 300 proteins co-profile with the whole transcriptome spot by spot across a variety of mouse tissue specimens and also in the human tissue, in this case, a very interesting tissue sample. We look at sort of the skin biopsy from Moderna vaccine injection site. We can see very early on the immune cell activation in the peripheral skin tissue region actually kind of correlates with systemic immune response later on. And so beyond the spatial analysis of gene expression and a panel of proteins, what we can do, we were very interested in sort of the mechanisms that control the expression of the genes, such as at epigenetic level. So those papers have been published. It just kind of highlight what we can do here to map down the chromatin accessibility in both mouse and human tissue specimens, and also the specific histomodification states like the different histomarks, K27 trimethyl, K4 trimethyl, and the K27 acetylation, even though you're now doing kind of multiplex spatial histomodification sequencing, but from the serial tissue sections, you can actually see reasonably well how the different histomarks work together in concert to control the development of specific tissue type or cell types. And you can further combine both, and the whole transcriptome and the whole epigenome pixel by pixel mapping over a very large tissue area, as demonstrated in this case for 3 different histomodifications and a whole transcriptome. You may ask, what's the added value once you have both? Of course, intuitively, that makes sense since epigenetics controls the expression of genes. You do want to find out the outcome or the output of that regulation, which is the expression of the genes, right? So it'd be nice you can do both. They can really compare. And so what you see here is the chromatin accessibility is a gene expression. In most of the cases, you do see pretty good concordance between accessibility and gene expression. But interestingly, if you look at, for example, this differentiation process from radioglia to post-mitotic premature neurons, and if you look at this differentiation trajectory based on RNA, based on ATAC, in general, you do see the chromatin opened up and the genes started to become expressed later. But that's not always the case for all the genes. For example, this group of genes, and the chromatin remains open, but those genes start express early on and quickly diminish. And so and then we look at what are that specific genes. Turns out those are not neuronal genes, that are kind of oligodendrocyte. So this is interesting once you have both modalities. The conclusion here is those cells, at least a subset of those cells, once they become committed to neuronal lineage, they somehow still maintain the epigenetic potential to become oligodendrocyte or other lineages at this stage of developmental process. And so now I would say this is spatial multi-omics, and hope you will join this hands-on computational data analysis session later on today, and you will get your own experience in kind of how to play with this spatial multi-omics data, including ATAC and the transcriptome and their combined data set. If I have a few more minutes, I want to highlight some latest development along this line in my laboratory. to map down spatial transcriptome, but not the standard spatial transcriptome you often see, which is just equivalent to spatial gene expression. So we were able to develop this methods using polyadenylation plus the Dibble chemistry to map down the variety of different RNA species, kind of total RNA species across the kind of large tissue, FLP tissue sections. This worked out so well for a variety of different tissue types. And in terms of data quality, we were totally amazed. And when you even compare our PathoDB to the probe-based FLP tissue specimen spatial transcriptome, we're able to get a comparable, even slightly better data quality in terms of gene count and the UMI count per spot. And but what is much more exciting is you are no longer limited to spatial gene expression. You can look at a lot of interesting RNA biology. For example, here, different alternative splicing isoforms, and as depicted by this diagram here, different isoforms we were able to detect spot by spot across the whole tissue section. Just to show you one specific example, this protein phosphatase subunit, and you see expression of this gene in multiple regions of the brain, at the isocortex, midbrain, and the dendrigyrus of the hippocampus. But this skippy isoform is exclusively expressed in the dendrigyrus of the hippocampus, but not anywhere else. So this type of information you just cannot get from spatial gene expression, spatial transcriptome data. So now we're able to see spatial alternative splicing mapping directly in clinical FLP tissue specimens. So similarly, we can look at other RNA species like small RNAs. We're able to detect in particular, the very challenging species of the RNAs and microRNAs, and which is typically 22 base pair long, and also from the same tissue, same spot, we have the whole transcriptome gene expression analysis result. So now you can draw correlation between every single microRNA and on the target genes, on the target transcripts to really understand this kind of microRNA messenger regulatory network. And another super exciting result from this spatial RNA biology profiling is you can look at the specific mutations. You can look at all kind of mutations, the single nucleotide variants across the entire genome scale. It's got on-target, unbiased mapping of mutational landscape. In other words, this is sort of like a tumor mutational burden analysis. As shown here, we just look at accumulated tumor, accumulated SNVs. You can see in a high SNV region really correspond to the malignant tumor cells as indicated by the specific staining used in pathology. If you look at all those mutations, perform mutation-specific clustering, untargeted clustering, you can even resolve the different subclones within the malignant cells based on mutational landscape. Okay, so I'm gonna stop here. I hope you appreciate how powerful the sequencing-based spatial transcriptomics spatial transcriptome and spatial multiomics can give you the huge power in discovery for cancer system biology research. I would like to acknowledge folks in my lab and all the amazing collaborators and the funding support, in particular, the Cancer System Biology Consortium. And thank you very much for your attention.